So just a very, very brief introduction on dendritic cells, because maybe people are not aware of these cells. These are cells of the immune system, and they are present in peripheral tissues where they sample antigens. And for instance, after an injury in the skin, for instance, dendritic cells can take up the antigens and undergo a process of maturation, and they migrate through the lymph to the draining lymph node, where they can present antigens to T cells and can initiate immunity to that given antigen. But dendritic cells can also migrate from peripheral tissue to the draining lymph node in a constitutive way, so in the absence of overt inflammation, and this is a process that can lead to tolerance towards self-antigens, and in the gut can be towards food antigens. Bacteria are entering the gut primarily via a portal entry, which is called M cells. M cells are scattered in what is called the follicle-associated epithelium, that is just lining the Peyer's patches. And here the bacteria then can be taken up by the dendritic cells that can migrate into the T cell area and can initiate an immune response. But I hope by the end of this presentation to show you that also dendritic cells play an important role in bacterial uptake. This is an image of the terminal ileum, and this is a Peyer's patch, and I should be starting. And here you can find, this is the lumen, this is where you can find food antigens, this is a Peyer's patch, and now you will see an area where you can find primarily B cells. These are the B cell follicles in the Peyer's patches, and the B cells are in blue. And then you can find an area where the T cells interact with the B cells. This is called the interfollicular region, and the T cells are in white. And then these are dendritic cells. You can see that dendritic cells are scattered almost everywhere. They are found primarily in the follicle-associated epithelium. But then if you look closely at dendritic cells, you can find sometimes these protrusions that go directly into the lumen. So now what are these protrusions? I just wanted to make an historical a story of how we describe these dendritic cells, because this was completely unexpected. And so, as many discoveries in science, you have to look carefully when you are looking at something, because sometimes you find something completely different. And then, as Philippe was saying, it's important to show what you are looking at, because seeing is believing. It's true. So everything started from this beautiful publication by Eric Plengol in Science, in which he was showing that M cells, the cells that I showed you are important for bacterial uptake in Peyer's patches. You could actually differentiate these M cells starting from enterocytes just by putting enterocytes together with lymphocytes, and primarily B cells. So if you had a monolayer of enterocytes, and you put them together with B cells isolated from the Peyer's patches, then these B cells would induce the differentiation of these enterocytes into M cells. And this would allow the bacteria to be translocated across this monocyte. So we wanted to know what was happening when we also added a second player, which was the dendritic cells, because as I told you, they are present in the Peyer's patches, and they are very close to the M cells. So we wonder whether the dendritic cells would also participate in this transformation. And so we just looked at the interaction between dendritic cells and epithelial cells in the presence or absence of bacteria. And what we found was that indeed dendritic cells were allowing the translocation of bacteria from this apical side into the basolateral side. And it was not dependent on the pathogenicity of bacteria, because both Salmonella typhimurium and Streptococcus gordoni, which is a commensal bacterium in the oral cavity, could allow the differentiation of these we thought at that point that it was a differentiation of enterocytes into M cells. But then, when we looked closely by confocal microscopy at these filters, we found that these dendritic cells actually were crossing the filter and were going through the epithelial cells, and were actually contacting directly the bacteria from the luminal side. Now the bacteria are in green and the dendritic cells are in red, and you can see here that the dendritic cells are contacting the bacteria, and they are also in the process of internalizing the bacteria. So it was completely different from what we expected. We were expecting these epithelial cells to transform into M cells, but actually we found that the dendritic cells were crossing the epithelium and were interacting with the bacteria. And when we looked at transmission electron microscopy, we could find these processes of dendritic cells in between the epithelial cells. And actually, they were establishing tight junction-like structures with neighboring epithelial cells. And I don't know how much you're familiar with tight junctions, but tight junctions are made of several transmembrane proteins like occludin, claudin-1, and JAM1. And we found that dendritic cells expressed all of these tight junction proteins, and this was a way for them to allow these tight junctions-like structures between them and the epithelial cells. So when we blocked JAM1 interaction between dendritic cells and epithelial cells by using neutralizing antibody to JAM1, we're also inhibiting the translocation of these dendritic cells. So these are dendritic cells. When you put LPS or Salmonella from the apical side, they're induced to move all the way to the apical side. By contrast, when you first incubated the dendritic cells with the anti-JAM1 antibody, you were also blocking their ability to go to the apical side, so suggesting that these tight junction proteins are involved in the process of the dendritic cell extensions. So then we looked in vivo, because this was just an in vitro observation, and we found that also in vivo, after Salmonella infection, after 30 minutes of Salmonella infection, we could see these processes. Some processes were going from one villi to another one. Some dendritic cells were found actually outside of the villi, and some processes of dendritic cells, now in red, here they were in green, they were contacting the Salmonella, which is in green. But again, these were static images. And even though we managed to publish this paper quite well, the scientific community was very skeptical about it. And not many people were really believing in this discovery. So first of all, there was another paper in the literature by the group of Hans Christian Reinecker in Science that described the same type of features, and this time they used a mouse that was expressing GFP and CX3CR1. CX3CR1 is a receptor for fructalkine, and they were showing that mice that were lacking CX3CR1 also lacked this extension of the processes. So the first information was that someone else in the scientific community could reproduce our data, which was good, but still it was a static image. So what we decided to do was to really look whether in vivo you could follow the extension of these dendritic cells. And so we switched from confocal microscopy to two-photon microscopy. The advantage of two-photon microscopy was already described by Nancy. The advantage is that you can, in a confocal microscopy, you have a pinhole, which is just in front of the detector, which is determining the optical section and the thickening of the optical section. By contrast, in two-photon microscopy, the two photons need to interact with each other in order to be excited and to release light. And so in two-photon imaging, the excitation occurs only at the plane of focus, so where the two photons really match with each other. By contrast, in the confocal imaging, excitation and then also bleaching occurs throughout the significant portion of the tissue. Moreover, when you have two-photon microscopy, you can go much deeper into the tissue because it's easier to get into the tissue by these two photons. And so what's happening is that you can go deep and you can analyze a good portion of the tissue, and because you have less photobleaching, you can do that also in vivo in an animal. For instance, these are the intestinal villi, and you can see that you can go much deep into the tissue, and you can follow several markers. This is just static images, but you can do that also in vivo. And to do that, you can do this directly into the mouse system. So what we used was a mouse in which all the MHC class II bearing cells were also positive for GFP, because this was a fusion under the MHC class II promoter. And you could see all the dendritic cells green because they were expressing MHC class II. So then you can give orally the bacteria, for instance, green Salmonella. red salmonella, otherwise with the GFP you cannot really follow them. And you can anesthetize the mouse. You can do everything in vivo, in the living animal, but he's sleeping, of course. And then with the 2-photon, you can go deep into the tissue, or you can create a hole into the tissue, and you can image what's going on there. So we did that in the terminal ileum, and you can go through the whole tissue, scanning the whole tissue with the 2-photon, and then you have a lot of zeta stacks that you can, in time, you can pile each zeta stack to have a 2D image, and then over time you can make a video. And so you can follow, because the dendritic cells are green and the salmonella is red, the interaction between the dendritic cells and the salmonella. So this is just a 3D reconstruction in order to show you how the dendritic cells are in a 2-photon microscopy image. So you can see these dendritic cells that are green, and now in red is the epithelium, because it's colored with NARF. You can see that these balloon bodies of the dendritic cells are present primarily at the tip of the villi. And this is where they most likely can interact with bacteria, because there is less mucus in this area of the villi. And you can see here by 2-photon microscopy that you can completely follow these extensions of the dendritic cells and this balloon body shape that they have. So now you can say, so how can you measure the amount of balloon bodies that you have across the vilum? So this is the tip of the vilum, and you go down, and you can measure the amount of balloon bodies that you have, and you can quantify and analyze their distribution. And you can see here that primarily at the tip of the villi you can have these balloon bodies that are present where they can most likely interact with the bacteria. And as I told you, here there is less mucus, because it's always washed away a little bit by the movement of the peristaltic movements of the gut. And also the bacteria are less found in the lower regions of the villi towards the crypts, because here you have lots of panel cells that can release defenses, which are antimicrobial peptides. And so this is probably why you can see these protrusions primarily at the tip of the villi. So then the question was exactly whether these protrusions would interact with the bacteria in vivo. And so here again the DC protrusions are in green and the bacteria are in red. You can see that there is a very long-lasting interaction between the dendritic cell protrusions and bacteria here and there, and it can take also 10 minutes of interaction. And if you look at this balloon body here, you can also see that these balloon bodies are not only interacting with the bacteria, but also internalizing the bacteria. And you can see that if you make this balloon body transparent, you can find a bacterium inside, so suggesting that these balloon bodies are not there only for interacting with bacteria, but also for actively capturing the bacteria in the intestinal lumen. So what drives the extension of the disease? Because I told you that they are present primarily at the tip of the villi, it's where they can interact with bacteria. So what we did was we treated the animals with antibiotics, and we looked at whether the number of the extensions was decreased, because if it is driven by the presence of the bacteria into the lumen, you would expect to see a decrease. And indeed, this is exactly what we see, is that this is in the proximal ileum, so the upper part of the ileum in the small intestine. You can see that there is a decrease by giving broad spectrum antibiotics. And it's also interesting that the number of the extensions of the disease depend on the region of the intestine that you're looking at, because if you look at the terminal ileum, you see less constitutively expressed extensions, but this can be induced just by giving salmonella orally. You can have a higher number of the extensions, whereas in the proximal ileum, there is not much difference. So you already have a high number of DC extensions in the untreated animal, and then there is no further increase after salmonella infection. So since bacteria seem to be involved in driving the extensions of the disease, are toll-like receptors involved in this process? So toll-like receptors are receptors that will recognize components of the bacteria. So the question was whether these were involved in driving the extensions of the disease. And you can see that if you while you're doing the microscopy, if you give toll-like receptor ligands like a peptidoglycan, lipopolysaccharide, which is a component of the gram-negative wall, or CpG, which is depicting bacterial DNA, you can see that the number of the disease is induced, DC extensions is induced, so suggesting that toll-like receptor ligand can indeed provoke the protrusions of the dendritic cells. So there are two possible mechanisms for driving the DC protrusions. One is a direct mechanism in which either bacteria or bacterial ligands can interact after transcytosis with the dendritic cells from the luminal side, and these dendritic cells are induced to transmigrate. Or a second possibility is an indirect mechanism that is mediated by epithelial cells. So the TLR ligands or the bacteria do not interact directly with the disease, but only with the epithelial cells, which is the first barrier. And then these are then translocating the signal to the dendritic cells, and then the dendritic cells are going through and actively protruding these extensions. So in order to study this, we looked at, we did chimera experiments, bone marrow chimeras. So we took mice in which either the TLR4 was knocked out, and these mice were literally irradiated, so the bone marrow cells were killed and were reconstituted with the bone marrow from mice in which all the dendritic cells would be green because they are from CD11C GFP positive mice. And then in this case you have dendritic cells that are wild type, so they do express the toll-like receptor 4, while the epithelium will be knocked out because the recipient mouse is a knockout for TLR4. And we did the same for TLR2 and for MYD88, which is a common adapter for all toll-like receptors except TLR3. And what we found is that indeed the expression of toll-like receptor by epithelial cells, but not by the disease, was involved in driving the extensions of the disease. Because if you take the chimeric mice in which the epithelium is knocked out for MYD88, but the dendritic cells are wild type, then you don't see anymore the induction of these extensions after Salmonella infection. By contrast, if you take, the same is true for TLR4 and TLR2 knockouts. If you take the whole bacteria, you still see the extensions because you have other toll-like receptors that could compensate for this. By contrast, if you give just LPS or peptidoglycan, you don't see the extensions of this disease. So I just wanted to conclude, and I hope I have convinced you, because seeing is believing, that dendritic cells can extend protrusions across mucosal surfaces, and these protrusions are involved in bacterial uptake, and that the extension of the protrusions depends on the presence of bacteria. So if you give antibiotics, you also reduce the number of the protrusions. And finally, that the extension of the protrusions depends on the expression of toll-like receptors on the epithelial cells, which is fundamental because these are the first barrier that will see the bacteria or the ligands on the luminal surface. And then finally, I wanted to acknowledge my collaborators, and in particular, Marcello Chiepa, who was first in the lab of Ron Germain, who did the work on two-photon microscopy, and now is back in my lab, and Monica Rimoldi, who was a technician in my lab, and now she's at Humanitas. And I wanted to thank you all for the attention.
Super, grazie Maria. Questions?
Yeah, Maria, first regarding the involvement of the chillers, I'm a bit surprised because, I mean, maybe I'm wrong, but if I remember well, they are not supposed to be so well expressed at the apical side of the epithelial cells. It seems that they are. And the second point is, what about commensals?
Yeah. Well, actually, it's quite controversial whether they are expressed or not on the cell surface. It's true that when we give the components, they could be transcytosed and go to the basolateral membrane. So we don't know whether they are acting from the apical side or from the basolateral side. But it doesn't seem that there are no toll-like receptors on the apical surface, but there is a disproportion, so you have more on the basolateral side. And regarding the commensal, so I think it's interesting the fact that in the proximal ileum, you already see the extensions of the disease. It's where you have less commensals, but still they're playing a role, because when you give antibiotics, you're reducing the number of the protrusions. By contrast, in the terminal ileum, where you should see more commensals, then you see less protrusions, and we still don't know whether this is a mechanism of tolerance induced by these toll-like receptor ligands, because you know that many cells in the gut, like including dendritic cells, they do not respond to toll-like receptor ligands because of the tolerance induction. So we don't know.
So I'm wondering, what do you think is biological significance of these projections? Because if you require a toll receptor for the induction of these things, I mean, we and others have shown that the toll-2, the toll-4, the toll-2, toll-4 double knocker, and the MYD8 is actually more susceptible for infections, and Salmonella has no problem in reaching the primary lymph nodes in these mice.
Yeah.
So how would you put it? So do you think it's important for presentation and not for entry?
In the beginning, I thought that these dendritic cells were primarily involved in tolerance induction. So they were there, they were taking up commensals all the time, and they were inducing tolerance. But then I don't think it's the case anymore, because they don't express these cells that are protruding, they don't express CD103, which is the marker for the tolerogenic dendritic cells. By contrast, it looks like these dendritic cells are the ones that are inducing T helper 17 type of responses. So it seems like they are there to constitutively produce a little bit of IL-17, which is probably involved in keeping constant the numbers of bacteria present in the gut. But we don't, I mean, this is just speculation.
Maria, do you have any idea about the range between, you have this, as I would say, at the steady state level, you have the DCs are somewhere. So, and then in a way they have two steps to go further. One is to really get as close as possible to the epithelial cell, basolateral side. And then they also have to move these projections in between the cells up to the lumen. And so this is, in a way, mechanistically represent two steps. And if you start thinking in terms of gradients of messages, for instance, you would need something that attracts them as close as possible to the disease, to the epithelial cells, but you would also need a second gradient which would attract the pseudopods. And this would have to come from the top of the epithelial cells. Like, Madara has been working on this for polymorphonuclear cells. Have you started to think about this sort of dual signaling that is required?
Yeah, we never really studied that, but it's really interesting. At some point I thought that maybe defenses would play a role, because CCL20, which is the chemokine that is attracting dendritic cells, that should express CCR6, which is not the case of these cells, because they do not express CCR6. But just to make an example, CCL20 is very similar to MIP3 beta. And so I thought maybe some other defenses might have a chemokine-like activity on the disease protruding. This would explain why you need bacteria from one side. And but this is something that we really never looked at. And also something that would be very interesting to look at is whether then the protrusion of the DC was polarized like it is the apical surface of the epithelial cells. This is also something that would be interesting to analyze, whether there you have something that it's more likely to be important for the interaction with bacteria or not.
Okay, thank you. All right, so if there are no more questions, thank you very much. Oh, sorry.
Sorry, it was just a short question. On the JAM molecules, which is involved with this extension, do you know which chyme is involved and whether you see a default of of capture of bacteria in JAML knockout, JAM knockout.
The JAM is exactly JAM A, and we actually always wanted to look at the knockouts because we have them in the lab, but we never looked at them. So we should do that.